that was the aha when this all happened was, okay, Robert, you got this whole new idea of using RNA as a drug, as an alternative gene therapy, and it has the advantage that it only lasts for a few hours. The RNA degrades. So you can treat it more like a drug. And if the patient has an adverse event, you just stop administering the drug. What's not to like? And what are you going to use it for? You can use it to produce an immune response. You can use it as a vaccine. Well, the thing about a vaccine is that you don't need very much antigen. So the fact that it's relatively inefficient suddenly becomes okay. But if you take a highly potent, biologically active protein like spike, you can amplify the immune response to it, and you can also produce a variety of adverse events associated with it. And then you combine it with this pseudouridine, for which the Nobel Prize was given. And that's an important one to unpack too. So Carrico and Weissman, I knew Katie Carrico. She objects to my using this term. I could say I coached her or mentored her or whatever. I gave her ideas. She called me early on and asked for some advice about how to get involved in the RNA world. And I introduced her to some people, and I talked to her about some of the key issues, invited her to a conference. And then this is all a decade after I'd done that stuff at the Salk. And her and Drew Weissman observed that a fundamental problem associated with this was the inflammatory response associated with the lipid nanoplex. And they inferred, and correctly, that a foreign RNA itself evokes an inflammatory response. This gets back to your fundamental question, along the lines of, can this ever be made safe and effective?